Welcome to Tomorrow Unveiled, hey. We are diving headfirst into what I think many are calling the most consequential single week in Geroscience this year.
It really was an incredible 7 days.
We're looking at the week spanning December 16th through the 23rd, 2025. And it was a period that delivered this stunning convergence of longevity breakthroughs across multiple disciplines. Our mission today isn't just to list these findings. We wanted to distill the functional imperative behind them. The goal of longevity research has, it seems, definitively shifted.
Oh, absolutely.
It's away from merely adding years to life and toward adding functional, vital life to those years. We're calling it the Healthspan imperative.
And that shift is not just, a change in words. It's absolutely pivotal. For decades, traditional medicine and even early longevity research, they treated aging as the series of inevitable separate diseases.
Right, you fought heart failure over here, then Alzheimer's over there, then maybe cancer.
Exactly. You were just fighting these individual battles in isolation. What our compiled sources are showing us now is that longevity science has fundamentally moved past that sort of incremental phase.
Okay.
We are now targeting the foundational biological mechanisms of aging itself. The root causes, and this is the critical part, we are translating those molecular mechanisms into measurable, tangible results.
Results for functional health span extension.
Right. This week really proves we are finally bridging that gap.
Okay, let's unpack this with a phrase that kept showing up in the materials our team pulled together, the Great Convergence. It seemed to highlight that these fields that used to be in their own silos, immunology, metabolism, Senolytics, even artificial intelligence, they're all kind of collapsing into one integrated field.
They are, because they all touch upon the same fundamental drivers of cellular decline. It's no longer about, one drug doing one thing.
It's about systematically repairing the entire biological system at once.
Precisely. And that means the research isn't fragmented anymore. Our analysis of the consensus identified 3 major, highly corroborated themes that really represent this convergence.
And what are they?
First, the resurgence of mitochondrial rejuvenation, basically fixing the cell's power plant at a structural level.
Okay.
Second, the maturation of precision Senotherapeutics. So moving from these broad-spectrum anti-aging drugs to highly targeted strikes against specific disease tissues.
And third.
And finally, the rise of AI as an aging measurement tool, which gives us that objective, real-time feedback loop we need to prove these interventions are actually working. We're going to unpack what these changes mean because they fundamentally redefine what an intervention could look like for you.
That sounds like an exhausting but exhilarating 7 days for Geroscience. Let's start right in the literal engine room, because if we want to talk about fixing the root cause, we have to talk about mitochondria.
We do.
Our first segment is part 1, the engine room update. Mitochondrial efficiency as the master clock.
The central role of mitochondria, the power generators of the cell, has been recognized for decades. But for a long time, the dominant theory, the mitochondrial theory of aging, it was pessimistic.
How so?
They were seen as these fragile batteries that just inevitably accumulate damage. This leads to oxidative stress and dysfunction, and it was just something you sort of passively watch degrade.
And the research from this week just flips that whole narrative completely.
It does.
It suggests we aren't just stuck passively watching them degrade. We can actively tune their efficiency, optimizing them to run cleaner and harder, almost like a mechanic can upgrade an old but still valuable engine.
That analogy of a tune-up is spot on, but we're really talking about a structural overhaul. Think of the cell as a high-performance sports car engine.
Okay.
In use, it's pristine. It's producing maximum horsepower. That's ATP with minimal exhaust. But in aging, the engine gets dirty. It produces less power because the components are misaligned, and it produces way more toxic exhaust, which we know as reactive oxygen species, or ROS.
So these breakthroughs are focused on structural repair.
Exactly. Making that engine efficient again to reduce the systemic toxicity that's driving all this age-related decline.
This is where it gets, I think, really fascinating and pretty technical. The research our team compiled detailed findings from researchers in Tokyo that confirm the critical role of a protein called KOX7RP.
Right. Cytochrome C oxidase subunit 7A related protein.
It acts as a master regulator of this specific mitochondrial efficiency.
This is a really deep dive into cellular architecture, and it changes how we think about the entire process. KOX7RP is it's essentially the scaffold or the assembly factor that stabilizes these massive interconnected protein structures.
The supercomplexes.
Also known as respirasomes, yes.
Wait a minute. I thought the process of cellular respiration relied on four separate complexes, IT3 and 4, just passing electrons along the inner mitochondrial membrane. What are these supercomplexes and why is their stability managed by this COX7RP protein so crucial for extending a healthy lifespan?
You're absolutely right about the individual complexes, but the supercomplexes are the organized macrostructure. It's the difference between a bucket brigade passing water from person to person and a fully enclosed high-pressure pipeline.
Ah, I see.
If the four complexes are just floating separately in the membrane, the electrons have to be ferried between them by these mobile carriers, specifically ubiquinone and cytochrome C.
And that's not efficient.
It's not. Every time an electron transfers, there is a risk it leaks out and creates that toxic exhaust, the reactive oxygen species. This electron leak is a foundational driver of oxidative damage.
So when COX7RP builds and stabilizes these supercomplexes, literally bolting those four components together, it creates that direct streamlined pipeline you mentioned.
Exactly. It ensures direct electron channeling. That process maximizes ATP production because the energy transfer is optimized, and at the same time, it dramatically minimizes the ROS leakage because the electrons are fully contained within this respiratory chain architecture.
So the engine becomes both more powerful and way cleaner.
Significantly more powerful and vastly cleaner. This discovery is so compelling because it focuses on preventing damage at the earliest possible structural point.
And the practical take-home from this is huge. The research showed that in mouse models, actively boosting this COX7RP expression resulted in a 6.6% extension in median lifespan.
Yeah.
How does that 6.6% benchmark against, say, the early results of rapamycin in mice, or even caloric restriction? Is this an outsized gain for targeting a single structural protein?
It's a very meaningful game, especially when you consider the precision of the target. Caloric restriction, or CR, that's kind of the gold standard intervention. It can yield maybe a 10 to 30% median lifespan extension.
It is hard to do it.
It requires huge behavioral changes and has some complex side effects. Rapamycin, one of the most effective drugs we know, often yields similar numbers, sometimes a bit higher.
So 6.6% is significant.
A 6.6% gain purely by optimizing a single structural protein within the energy production pathway confirms that metabolic efficiency is a master clock for longevity. It tells us that structural integrity is just as important as the chemical pathways we've been studying for years. It's an incredibly clean result.
A small, precise change yielding a systemic, measurable difference.
Exactly.
And it wasn't just about survival, which is important, but about health span. What were the specific systemic benefits they saw in those mice?
The data showed that fixing the engine globally benefited the entire organism. The COX7RP-enhanced mice showed significant improvement in glucose homeostasis.
So better insulin sensitivity.
Much better. And much less metabolic stress, effectively combating the kind of type 2 diabetes pathology you often see in aging models. They also had dramatically lower blood lipids and total cholesterol, suggesting superior cardiovascular health.
What about physical performance?
They demonstrated enhanced muscle endurance, which is a key measure of frailty. And crucially, at the cellular level, they showed lower levels of systemic inflammation and a significant reduction in markers of cellular senescence.
So by tuning this one structural piece, they systematically modulated 3 or 4 of the hallmarks of aging all at once.
That's right.
That's really robust basic research, and it points the way toward highly specific drug targets. But the story doesn't end there. Our sources also show a critical piece of clinical validation this week, moving from mice to a measurable human impact with urolithin A.
Yes, and this transition from why it works to does it work in people is what makes this week so important.
Right.
This brings us to the Mitoimmune study, which validated urolithin A. It's a compound produced when specific gut microbes process dietary polyphenols.
The kind you find in things like pomegranates, right?
Exactly. It was validated as a potent anti-aging intervention. Now, the mechanism here is a little different, but completely complementary to COX7RP. If COX7RP prevents the damage, urolithin A cleans up the damage that has already happened.
Because urolithin A induces mitophagy. For our listeners, we know this is a core cellular maintenance process, but let's just refresh its specific importance here.
Mitophagy is the cell's internal quality control and recycling program. It's this highly selective process of identifying, tagging, dismantling, and then recycling only the old, damaged, debris-filled mitochondria.
And then it uses the parts to build new ones.
It does. Once the components are recycled, the cell uses them to build brand new, pristine power generators. So if COX7RP is the architect stabilizing the new engine parts, urolithin A is the maintenance crew clearing out all the junk.
And the research team connected this regenerative process to a specific age-related crisis, immunosenescence, the aging of the immune system.
Yes.
Why does clearing mitochondrial debris matter so much for the immune system? Maybe more so than, say, a skin cell.
This is maybe one of the most direct and functional links between cellular aging and the collapse of health span. As we age, our adaptive immunity, our ability to mount a defense against new pathogens or respond to a vaccine, it starts to fail.
And T cells are the key here.
Our T cells, the specialized, energy-intensive warriors that fight infection and cancer, are the first to falter. And the reason why is because T cells are hyper-energetic. When they get the signal to activate, they need a huge, immediate burst of ATP production. They have to go from 0 to 60 in a second.
And if the batteries are old and clogged,
They stall. If their mitochondria are old, debris-filled, or just inefficient, the T cells fail to proliferate or activate correctly. They can't spring into action to fight an infection.
which is why older adults have poor vaccine responses.
And are more susceptible to severe illness. What's worse is that these stalled, energy-depleted T cells often become dysfunctional and start leaking inflammatory signals themselves, which contributes directly to chronic low-grade inflammation or inflammaging.
So the immune system basically quits, not because it lacks the instruction, but because it lacks the energy to execute that instruction.
Precisely. And the breakthrough in the human mitoimmune trial was showing that urolithin A successfully cleared mitochondrial debris specifically in the immune compartment. This led to a functional restoration of T cells.
They acted like younger cells again.
They could activate and proliferate like younger cells, and there was a corresponding reduction in systemic inflammatory markers in middle-aged adults. This wasn't just a mouse model finding, it's a critical, confirmed human bridge across that translational gap.
So a validated, druggable way to clear damage and restore function in humans. That is the 1-2 punch we needed.
It confirms metabolic efficiency is a primary target.
Okay, so if mitochondria are the faulty engine, our next topic, senescent cells, are the toxic waste that engine leaves behind. Let's transition to the next big theme of the week, part 2. Precision Senotherapeutics, the targeted strike on zombie cells.
This segment really marks the maturation of the whole senolytics field. I mean, the early search was for a single, broad compound, a pan-senolytic to clean up the whole body.
But that's evolving.
It's evolving. The sources we looked at highlight this necessary and really exciting move toward highly targeted precision warfare against specific types of senescent cells that drive specific diseases in specific organs.
And this focus on specificity is key.
It's key to maximizing efficacy. And, just as importantly, reducing the potential for off-target effects.
We should quickly establish the baseline here. I think our listeners are familiar with senescent cells, but let's be really clear about their primary threat.
They are the zombie cells. They're damaged cells that have stopped dividing, but crucially, they refuse to undergo programmed cell death, or apoptosis.
They just hang around.
They hang around, and the primary threat isn't just their presence, it's what they secrete. They emit this toxic cocktail of inflammatory factors, known as the senescence-associated secretory phenotype, or SAS.
And the SASP poisons the healthy tissue around them.
It poisons surrounding healthy tissue, driving chronic inflammation, tissue breakdown, fibrosis, and dysfunction across multiple organ systems. So eliminating them is fundamental to combating several hallmarks of aging at the same time.
Okay, now for what I thought was the most surprising finding from the research this week, the application of Senolytics for brain health, specifically targeting an epilepsy link.
Yeah, that was a big one.
Most people think of Senolytics for, painful joints or lungs or heart disease, not drug-resistant neurological disorders.
This is a major paradigm shift because it reframes a chronic brain disease as an aging disease. One driven by the tissue environment, not just by neuronal excitability. Researchers looking at drug-resistant temporal lobe epilepsy, or TLE.
A very difficult condition to treat.
They found strong evidence suggesting that TLE is actually a pathology driven by senescent glial cells.
So not the neurons themselves, but the brain-supporting cast, the astrocytes and microglia. Why are these support cells turning into zombies? And why does that cause seizures?
Well, the hypothesis, which is now supported by this evidence in animal models, is that often following a brain injury or even just chronic stress, these glial cells can age rapidly and become senescent.
And they don't just sit there quietly.
No, when they turn into zombie glial cells, they actively secrete huge amounts of those inflammatory SASP factors, things like IL-6, IL-1 beta, and specific chemokines.
And these factors irritate the neurons nearby.
They chemically irritate them, creating this constant state of low-level stress and hyperexcitability. This constant irritation effectively lowers the seizure threshold, making chronic drug-resistant seizures almost inevitable.
So the focus should shift. Instead of just trying to suppress the neuronal activity with anti-epileptic drugs, which often fail, you clear the inflammation in the surrounding tissue that's causing the problem in the first place.
Precisely. You're treating the root environmental driver. So the research team cleared these senescent glia in animal models using a classic senolytic combo, dasatinib and quercetin.
Thank you.
And the results were profound. Not only did seizure frequency plummet by up to 80%, but the therapy even rescued cognitive function.
So it reversed memory loss.
Memory loss, which is often a devastating side effect of chronic TLE, was significantly reversed. This suggests the memory impairment was a direct toxic effect of these SASP factors, not just collateral damage from the seizures themselves.
That opens a massive, immediate therapeutic avenue.
Right.
40% of epilepsy patients are drug-resistant. This could be the first real disease-modifying therapy for millions of people.
It's a huge translation from a cellular concept to a devastating human condition.
Okay, let's look at how researchers are making these senolytic strikes more surgically precise. The sources detailed a fascinating new technological approach called the Feroptosis bypass. It sounds like it relies on a clever Trojan horse strategy.
Specificity is the critical hurdle. As you said, senescent cells are incredibly hard to kill because they've locked themselves down. They are masters of survival. They have these robust anti-apoptotic defenses, built-in survival pathways that make them resistant to normal cell death signals.
You can't just tell them to self-destruct.
No. And if you use a broad-spectrum killer, you risk off-target toxicity to healthy non-senescent cells.
So how does this new Trojan horse tech bypass their internal anti-suicide defenses and achieve that perfect specificity?
It uses something called enzyme-activated prodrugs. This is the key to the Trojan horse. The drug is synthesized and given in a chemically inactive form. It only becomes toxic when it encounters an enzyme that is uniquely and highly abundant inside the target senescent cell.
It's like a molecular fuse.
It is.
And what is that highly specific enzyme that serves as the perfect target?
It's senescence-associated beta-galactosidase, or SA-beta-gal. Senescent cells express this enzyme at uniquely high levels. It's a consequence of their altered metabolism. So the prodrug is chemically masked, often linked to a galactose molecule.
So it's inert when it goes in.
Completely. But when the senescent cell internalizes this prodrug, its own high levels of SA-beta-gal cleave that chemical bond, activating the toxic payload specifically inside the zombie cell. Healthy cells have much lower levels of that enzyme, so they just leave the drug inactive and it harmlessly passes through.
That is truly precision molecular design. You're basically weaponizing the aging cell's own unique metabolic signature against itself.
That's a great way to put it.
And once it's activated, what does the drug actually do?
It triggers a non-apoptotic death pathway called feroptosis. This is an iron-dependent form of regulated cell death that's driven by massive lipid peroxidation.
So it rusts from the inside out, essentially.
You could think of it like that. The activated drug causes the cell's internal lipids to oxidize uncontrollably, leading to membrane rupture and a violent internal collapse. And because this mechanism is iron-dependent and completely separate from the normal apoptosis pathways,
The ones the senescent cell has locked down.
exactly, it entirely bypasses the senescent cell's anti-suicide defenses. This strategy is a huge leap forward in safety and specificity.
That is a remarkable combination of molecular elegance and functional effectiveness. But let's look at a critical functional application, particularly for frailty. The research highlighted an application for improving body composition and combating sarcopenia using a targeted senolytic.
This addresses one of the most visible and devastating aspects of aging. Frailty in the elderly often presents as sarcopenic obesity.
The vicious cycle of losing metabolically active muscle mass while gaining visceral inflammatory fat.
Right, and that combination severely limits mobility, increases inflammation, and predicts all sorts of poor health outcomes.
And the specific drug detailed, PTC-2105, produced results in aged mice that, I have to say, sound almost too good to be true.
The results were genuinely exciting because they suggested not just maintenance but active remodeling. This specific senolytic, PTC-2105, which is also mitochondrial targeted, achieved a remarkable 34% reduction in fat mass and a 23% increase in lean muscle mass in aged animal models.
Over a relatively short period.
Yes.
A drug that achieves significant muscle gain while simultaneously driving fat loss. That sounds like the definition of a Healthspan holy grail, especially since so many current weight loss medications risk muscle wasting.
It suggests something far more fundamental than just simple metabolic regulation. It indicates that clearing senescent cells, specifically those senescent fat cells and muscle stem cells, doesn't just stop decline. It actively removes the inhibiting inflammatory factors, the SASP, that were preventing local stem cells from doing their job.
So you clear out the zombies, and the healthy stem cells can repopulate the tissue.
You're essentially resetting the tissue composition back toward a youthful state. Senolytics are moving from being therapies for specific disease symptoms to being systemic rejuvenators that can combat frailty and reverse tissue composition.
The pace of scientific acceleration here is just undeniable. But if we're going to use these powerful tools in the clinic, we need objective proof that they're working. And that brings us perfectly to our third major theme, Part 3, the AI Conometer, redefining biological time and monitoring. We've covered some intense cellular architecture and drug mechanisms today. If you're enjoying this analysis on Tomorrow Unveiled, please remember that your support helps us continue to analyze these breakthroughs. If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future. Welcome back. So AI is no longer just processing the massive amounts of data generated by Geroscience. It's moving into the realm of perception, actively quantifying, and seeing biological time in routine tests in ways we never could before.
And this measurement transformation is perhaps the most practical advance we saw this week. For decades, longevity researchers relied on chronological age, how many birthdays you've had. And we know that's a poor, inconsistent predictor of individual health risk.
Right, two 60-year-olds can be in vastly different states of health.
Exactly. Now, AI is helping us refine and validate tools that measure biological age, the true age, and accumulated damage across your body's systems.
And the sources detailed a massive, global comparative study that validated the next-generation epigenetic clocks. What was the key consensus that emerged from this huge undertaking?
The study was ambitious. It compared over a dozen distinct epigenetic clocks against 174 different disease outcomes and mortality rates, tracked over a 10-year period in tens of thousands of subjects.
Oh.
And the consensus was overwhelming. The second and third generation clocks, the ones that incorporate phenotypic health data alongside DNA methylation, are vastly superior tools for clinical use.
Could you elaborate on that distinction? What makes clocks like PhenoAge, Grimmage2, and Dindon-Pace so much better than the earlier models?
Well, the early clocks simply looked for methylation patterns that correlated with chronological time. They were interesting, but they were pretty poor predictors of actual risk.
I see.
The next generation incorporates phenotypic data, things like creatinine levels, glucose, albumin, white blood cell counts into the methylation algorithm. They are essentially measuring the effect of aging on your body's function, not just the passage of time.
So they are far better at predicting actual disease risk and mortality.
Exactly. They link biological age acceleration. So the difference between your chronological age and your predicted biological age directly to clinically relevant endpoints like cardiovascular events, specific cancers, and all-cause mortality.
And Dindon-Pace is a bit different.
Dindon-Pace in particular is validated to measure the pace of aging, how quickly an individual is declining relative to the population. And this is absolutely crucial for tracking whether a drug like urolithin A or Senolytic is actually slowing down your personal aging rate. It closes the accountability gap.
So AI is turning our complex blood work and DNA into these objective, clinically meaningful risk scores. But the most stunning AI breakthrough mentioned was something called CXR-Age, the X-ray time machine.
This is a huge step toward the democratization of biological age assessment.
It takes it out of the realm of specialized DNA tests and into routine healthcare.
It does. Researchers developed a deep learning model that can predict a person's biological age and 12-year mortality risk with remarkable accuracy, simply from a routine, low-dose chest X-ray.
That's incredible. Think about how common that test is.
It's existed for over a century.
Wait, if a simple X-ray can predict my mortality better than my chronological age, that fundamentally changes how I'll view my annual physical. That's actually a little terrifying.
It is.
How is the AI possibly reading aging from a standard chest X-ray? It's not looking for obvious tumors or acute infections, is it?
No, it's detecting entropy, the subtle systemic decay that human radiologists are trained to ignore because it doesn't represent an acute pathology.
What do you mean?
When a human radiologist looks at the image, they're trained to spot a tumor or pneumonia. The AI, however, is trained on millions of X-rays to recognize the subtle, consistent changes that correlate with deep biological aging. It sees these nuanced shifts in the texture of the lung tissue, the stiffening and geometry of the aorta, the subtle loss of density in the bone structures near the spine and ribs.
It's seeing the gradual systemic decline that occurs across multiple organs at once, the general wear and tear that we just miss with the naked eye because it's not a single clear diagnosis.
That's the key. And this is revolutionary because it requires no new expensive hardware or invasive samples. Any clinic in the world with a working X-ray machine and a server can apply this algorithm.
It makes precision longevity assessment universally accessible.
It does. It expands the ability to assess biological age from these specialized multi-thousand-dollar longevity clinics to standard, even rural healthcare settings.
And that idea of constant, cheap accessibility is being amplified even further by consumer tech. The source material connected AI to wearable biometrics, leading to the PPH clock.
We're moving toward continuous passive monitoring providing real clinical meaning. The photoplethysmography, or PPG data, that's what your standard smartwatch or fitness tracker uses to measure your heart rate.
Those tiny pulses of light on the back of the watch.
Right, detecting blood flow dynamics beneath the skin. The PPH clock is an AI model that analyzes the pulse waveforms captured by these ubiquitous devices.
But given the variability and, let's be honest, sometimes lower fidelity of consumer wearables, how accurate is this PPH data in terms of clinical relevance?
Well, the analysis showed a strong correlation between these subtle pulse waveforms, which reflect arterial stiffness, vascular health, and blood flow and predicted biological age. Crucially, it was validated to predict cardiovascular events.
Okay, that's significant.
And what's more immediately useful for you, the listener, is that it tracks changes based on behavior. If you start sleeping better, consistently exercising, or you quit smoking, the PPH clock should show a corresponding measurable decrease in your biological age over time.
It turns passive, constant monitoring into a real-time precision longevity tool.
Exactly. It closes the loop between your behavior and a quantified outcome.
So we have the mechanical fix with mitochondria, the cleanup crew with Senolytics, and the objective speedometer with AI clocks. The science is accelerating incredibly quickly. But as these biological barriers fall, we have to pivot and confront the real-world hurdle that threatens to make these breakthroughs irrelevant for most people.
Yeah.
Let's pivot now to part 4. The Great Divide: Access, Ethics, and the Longevity Economy.
The scientific acceleration is undeniable, but the practical application is facing some very serious ethical and financial challenges. The stark question is, who benefits first?
And the sources documented a reality of stratification of access, the price tag problem.
It's already defining the field.
So give us the hard numbers our research team found. How much does cutting-edge longevity cost right now?
The numbers are staggering, and they demonstrate a distinct stratification in health access. Comprehensive longevity clinics, which offer full diagnostics, deep genome sequencing, advanced epigenetic clocks, detailed metabolic testing, bespoke supplement programs.
It's a whole 9 yards.
All of it. They can cost anywhere from $10,000 to over $100,000 per year. And that's just for personalized management and screening.
It's just the diagnostic package.
Yeah.
If you look at specific cutting-edge therapies, the costs escalate into a realm that's, well, unimaginable for the average person.
It is. For example, a single dose of the breakthrough gene therapy for sickle cell anemia, which is functionally a massive life extension and quality of life treatment, is priced at $2.2 million per treatment.
$2.2 million.
If that's the baseline for complex, curative, one-time interventions today, what will the cost be for systemic aging reversal therapies tomorrow?
That puts these life-changing therapies firmly in the realm of the ultra-wealthy. We're seeing this huge contrast between high-tech, ultra-expensive interventions and the low-tech, accessible ones.
A stark contrast indeed. You have affordable, repurposed drugs like metformin and rapamycin, which have shown benefits in animal models and are generic. They're economically accessible. But the complex, cutting-edge cellular, gene, and precision senolytic therapies are inherently limited to a very small affluent population. And this disparity raises fundamental questions about equity and the potential for creating a literal biological caste system.
Where Healthspan is dramatically determined by wealth.
Yes.
And the cost isn't the only hurdle. The regulatory and commercial landscape isn't catching up with the science. It creates this perpetual gap between what the lab knows is possible and what the doctor can actually prescribe and what insurance will cover.
That's the persistent insurance gap and regulatory bottleneck. The compiled research confirms that standard insurance, including national health programs like Medicare in the U.S. currently, does not cover most of these functional longevity interventions.
So that includes off-label use of promising drugs like metformin or rapamycin.
It does, and often even newer metabolic enhancers like the GLP-1 agonists, if they're used specifically for weight loss or longevity, rather than a pre-existing diagnosis of type 2 diabetes.
And the biggest regulatory blocker, the one that affects the entire drug development pipeline, is the FDA's fundamental stance.
Yes. The FDA currently does not recognize aging as a disease indication. This is the single largest, most fundamental barrier to progress.
Why is that so critical?
Because drug companies cannot conduct phase 3 trials explicitly designed to slow aging because there is no regulatory target. There's no recognized endpoint that says the drug works. This is why the TAME trial targeting aging with metformin is so important.
Right.
It doesn't aim to cure aging. It aims to treat a basket of age-related diseases simultaneously, hoping that success in that large combined indication will force a regulatory pathway open for anti-aging drugs generally.
So the regulatory framework is stuck in the 20th century, treating aging as inevitable, while the science is in the 21st, treating aging as malleable.
Exactly. Regulatory evolution, things like the proposed THRIVE Act, which seeks to modernize the FDA's view of aging, will ultimately determine the pace of translation. If aging were recognized as a treatable condition, it would unlock enormous capital for research and allow insurance coverage for interventions aimed at prevention, not just treatment of late-stage disease.
That regulatory shift could democratize access almost overnight.
It could.
Beyond the financial and regulatory challenges, the AMA Journal of Ethics addressed the profound societal implications of longevity this week, debating the civic virtue of life extension. This moves us from logistics into philosophy.
It does. The core debate, explored by bioethicists, centers on whether radical life extension for only a segment of society strains social bonds or creates cultural stagnation.
What do they mean by that?
Well, if the wealthy attain significantly extended health spans, living 30 or 40 years longer than others, does this strain the economic system? Does it impede the necessary social renewal that comes from generational change? Some scholars argue that passing the torch in leadership, culture, and business is an essential civic duty that long health spans could subvert.
But the counter-argument, which I've heard, is that extended health span in key sectors means accumulated wisdom and institutional memory, right?
Absolutely. The argument is that a leader, a scientist, or an expert who knows they will live for 150 years might make profoundly better, more sustainable long-term decisions, especially on issues like climate change or national debt.
Sure.
However, the immediate pressing ethical concern isn't abstract immortality or policy planning. It's whether the lag of equal access to these breakthrough therapies, the mitochondrial fixes, the precision Senolytics, will fundamentally widen the existing dangerous gap in health and vitality between socioeconomic groups.
And the data from the pricing structures suggest that disparity is already happening right now.
It is.
That is the crucial takeaway from this entire discussion. The science is moving at light speed, but the policy and access framework is lagging dangerously behind. If you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could take a moment right now to leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners and keeps us analyzing these vital source materials. Okay, let's bring all these threads together for the listener. After absorbing all the major findings of this pivotal week, what were the core corroborated takeaways that redefined where we are in geroscience?
I think this week was defined by breakthroughs that address the core mechanisms of aging simultaneously and with precision. It confirms we have fundamentally left the era of pure speculation. We identified 3 core advances.
Okay, what's number 1?
First, the mitochondrial fixes. The discovery of the COX7RP architecture dictating efficiency, and the clinical validation of urolithin A for functional human immune rejuvenation. These are moving mitochondrial repair from theory to measurable functional impact in both animals and humans.
Second, the Senolytics are achieving crucial precision.
Yes. They're moving beyond broad-spectrum joint repair to potentially treating complex drug-resistant brain diseases like epilepsy, using these highly sophisticated new mechanisms like the Feroptosis-based Trojan horse prodrugs.
They're moving from maintenance drugs to true disease-modifying therapies.
They are. And third, the AI and biometrics are providing the objective, real-time measurement and accountability we desperately need. Tools like CXR-Age and the validated epigenetic clocks are closing that translational gap, giving clinicians and researchers tangible, objective metrics of biological time that can prove an intervention is actually working.
The science is finally giving us a reliable map and a speedometer.
A great way to put it.
We love hearing your thoughts. Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about the ethical debate surrounding access to these incredibly expensive longevity therapies. Should governments intervene to democratize this life-extending science, or is it inevitable that the wealthy will always be the first beneficiaries?
As a final provocative thought, let's connect all this breakthrough science back to something deceptively simple: muscle.
Okay.
The compiled research noted a critical counter-effect of modern pharmacology. Rapid weight loss in the elderly due to GLP-1 drugs, which are otherwise excellent for improving metabolic health, often causes significant sarcopenia, muscle wasting.
Which is the opposite of a health span or longevity outcome, even if the patient is metabolically healthier.
Precisely. It highlights a conflict in priorities.
Yeah.
Short-term weight loss versus long-term vitality.
Right.
So if muscle is truly the primary organ of longevity, as the main repository of metabolic health, insulin sensitivity, and the critical buffer against frailty, this raises a fundamental question for the future of intervention. The question is not just how to lose weight or how to take a pill to fix cellular damage, but how to maintain high metabolic function and mass in aging muscle. Should we view fitness, nutrition, and resistance exercise not just as a lifestyle choice, but as the essential non-negotiable precursor that makes any future pharmaceutical longevity interventions safe and effective?
That's a powerful idea.
The path to a long, robust life may start not with a $2.2 million gene therapy, but with the prevention of frailty today.
A compelling challenge to leave with, shifting the focus back to the immediate and the actionable. Thank you for joining us for Tomorrow Unveiled. We'll see you next time.